High school. My name is Yassine Kawaji, and today we'll be highlighting CRM1 or exportin-1, a protein that in humans is encoded by the XPO1 gene. The nuclear pore complex is a large complex embedded within the nuclear envelope that plays a prominent role within the cell. This complex is responsible for importing and exporting proteins as well as RNAs between the nucleus and the cytoplasm.
So CRM1, the protein that we choose to model in this structure right here, is able to drive chemical reactions, and it also binds to protein cargos, which allows for transport through the nuclear pore complex. So here I have the structural ligase, and this is specifically important because it can bind to the active site right here. So it can bind here in the protein. Yeah. So then CRM1 is composed of 21 tandem heats, which is an acronym for the proteins it's composed of, as well as the hydrogen molecules and the alpha helices and beta plated sheets as shown in the molecule here. And the structure of CRM1 allows for these processes to occur, and the plasticity of CRM1 also aids in the binding of also aids in the transfers of proteins between the nucleus and the cytoplasm.
So CRM1 acts as both an importin and exportin. It drives chemical reactions and binding of protein cargos for transport through the nuclear pore complex. When there is an overexpression of CRM1, it stimulates Rev which promotes unspliced RNAs and USN RNAs, which is exported from the nucleus. Therefore, when leptomycin B binds to CRM1, the nuclear exports of Rev and USN RNAs are inhibited. So for the nuclear import within CRM1 is that the important proteins bind to the cargo in the cytoplasm after they interact with the nuclear core complex and pass through its channel. Once it interacts with the RAN GTP, there is a conformational change that causes it to dissociate from its cargo. The resulting complex translocates to the cytoplasm, where RAN binding protein, or RAN BP, separates RAN GTP from its important. Separation allows for hydrolysis to be induced by the GTP-ACE activating protein.
Nuclear export is mediated through the chromosomal region by the protein CRM1. In the absence of an inhibitor, the binding groove remains closed, but in the presence of one, such as leptomycin B, as previously spoken on by members of this group, it opens in order to bind to its nuclear cargo. Covalent conjugation is the process responsible for the opening and closing of the groove, which varies due to rearrangement of amino acid side chains, such as the two that are highlighted in our model. In red and blue, there are cysteine and arginine respectively, as well as the orientation of the helices. So RAN GTP, which is the beige structure in the photo that you see on the board, is another protein involved in the process that is responsible for the hydrolysis that leads to the restored complex that contains a restored nucleus. And the main idea, big picture, if you will, is for the mRNA to be able to leave the nucleus freely, which leads to the development of proteins, which we need to function. So without this process, nobody would be able to sit here and observe this presentation as you are right now.
This is made possible by the Camille and Henry Dreyfus Foundation, a special grant program in the Chemical Sciences and HHMI pre-college program. And a special thanks to Dr. Natalia Creighton, Dr. Garbarino, and Shannon Colton, and an immense thanks to our teacher, Ms. Ross.